So for chapter 4, it focuses on innate immunity, and so it builds on the information we already know from chapter 1, where we were given an introduction on innate immunity and adaptive. In chapter 2, we talked about different cells. Some of these cells play a role in innate response, and then chapter 3, we looked at some of the receptors that are present on innate cells. And so again, this just goes into more detail than those previous chapters. So one of the useful figures from this chapter or table is the table you're seeing here. We're going to, in chapter 5, when we look at the adaptive, look at the right side. But for this chapter, for this audio lecture, we'll focus on just the innate. So as we talked about before, the innate response is quick. So within minutes to hours after exposure to non-self. With specificity, it's not specific to a specific molecule-like sequence, but it will recognize patterns. So it's specific in the fact that there's these patterns there, and they're from pathogens. They're not from your own cells, or they're produced by your damaged or dead cells. So cells, your own cells, are able to produce these danger signals, and these danger signals tell the immune cells, the innate immune cells, to engulf and get rid of. Okay, so it is more limited, as we talked about. So there has to be the germline coding. So from the DNA coding, there's not all the rearrangement that happens that we see with the adaptive cells, and we'll talk about later on in the semester. And there's not the same memory that we see with adaptive. So we think of a memory being from the adaptive. There is some, because there are some specialized cells, the residents, some of the natural killer cells are able to, I think of it, they've received more education, so they're more mature, and so their response is slightly different than the immature or non-resident free cells. Okay. With innate, we have to be able to distinguish between self and non-self, and again, it's through these molecular patterns that are found, the MAMPs that are found, and through the pattern recognition receptors, okay, that they're able to distinguish between what's a microbe and what's self. Okay, and then when we look at components that are in the blood, there will be antimicrobial peptides. So you can think of these like little antibiotics that your cells will produce, your immune cells and your own cells will produce. And there can be other mediators such as cytokines that are going to be in the blood. And then you'd also have the cells in circulation in the blood, but those wouldn't be soluble. So that's why they're not on that row. When we look at the different cell types, phagocytes are going to be a general term, as we've mentioned. Different immune cells that are phagocytes would be your monocytes, macrophages, neutrophils, and dendritic cells. So all these can undergo the process of phagocytosis. I just want to point out that after these phagocytes undergo phagocytosis, they can become antigen-presenting cells in the adaptive immune response. So just keep in mind that later on, when we're talking about antigen-presenting cells, we're really talking about phagocytes. It's just their role has switched from functioning in the innate to functioning in the adaptive, but they're the same cells, but you have to have undergone phagocytosis to then present antigen. So again, just to kind of disclaimer, clarify that. We then have natural killer cells, NK cells, that are part of the innate. And then there's some additional leukocytes. So there's those innate lymphocyte cells. And then there's epithelial cells and endothelial cells can have function in the innate because they can produce interferon, and they can produce different signals that signal to the innate cells. So just looking at the innate response, we can break it down into 3 steps or stages. So we're going to have these physical and chemical barriers that prevent infection, right? So we have these epithelial layers of the skin. We also have these mucosal tissues, and those all create a barrier between the outside world and the inside world. So it protects, us inside from from potential exposures to pathogens by having, our skin. And we can see how if we have breaks in the skin or we've had burns, that we're going to be more prone to infections because this breaks that physical barrier. Okay, there's also going to be chemical barriers. So just if we think about some of the properties of our skin, it's going to have an acidic pH. It also has a higher salt concentration. So if you've taken microbiology, you're probably familiar with the mannitol salt agar plates, MSA plates. So those have a more acidic pH and a higher salt concentration. So those MSA plates are very much like the skin in a chemical sense. Okay, and so bacteria that can grow on the MSA plates can grow on your skin. So if you take an MSA plate, you would see things like Micrococcus luteus or Staphylococcus aureus or other types of staph present growing on that. And that's because they're able to grow in a more acidic environment. So yes, some bacteria can grow in this environment. Many of those are beneficial to us. But the majority of bacteria can't grow in these acidic environments. And so it actually limits potential pathogens, potential microbes from growing on your skin, so that it actually shifts it to more favorable ones. Your cells would also be producing, your epithelial cells would also be producing antimicrobial peptides and proteins. Again, these act like antibiotics. So instead of you having to take it, your cells make it. And that helps to damage and kill and harm those potential pathogens, so they're not going to, they're less likely to cause an infection. Okay, and then we have these cellular responses. So if there's a break in the physical or chemical barriers, maybe this is because we wash our hands too much. We're playing in dirt, or we're taking an antibiotic, or we again have a break in our skin, whether it's something like a splinter or paper cut or something more severe like we've had surgery done. If there's a breakdown in that first line of defense, the physical and chemical barriers, then that's where the cells, the cellular response to an infection can happen, right? And so this is where our macrophages and our dendritic cells are going to, jump into action, and they're going to undergo the process of phagocytosis, so that they are going to engulf and break down that material. We're going to have cytokine production. That's going to lead to inflammation. Additional antimicrobial substances will be produced, and so that the pathogen ends up getting eliminated. Again, we want to keep in mind that another part of the innate system, and what's in this figure shown as step 3, is activation of the adaptive immune response. So we have these phagocytes going to the lymph nodes, going to the spleens, where we're going to have T cells and B cells, so that we get a T cell response and an antibody response, right? So if we get that, splinter, and we might have some bacteria in there, we want to deal with that potential infection right away. And that's what innate does. But we also want to make sure that we bring in those lymphocytes, those T cells and B cells, so that they can respond, so the antibodies can be present, so that they can respond in case the innate system isn't able to deal with it, right? So innate's going to deal with it immediately, but we want to make sure we're already calling in the recruits. So again, the cytokine production, inflammation, all that helps to draw other immune cells to that location. And again, this could be free macrophages in circulation, or it could be T cells being called to that region. So a lot of information in that table in that figure. Okay, so again, keep in mind that you should know those 3 steps. So just looking at these 3 steps a little closer, as I mentioned, there's epithelial barriers that are going to create a physical barrier to prevent pathogens from entering into the body. And so most prominent would be the skin, because it covers you. And then we have mucosal membranes, right? So we have in the mouth, in the alimentary canal, we have mucus production. In the respiratory system, we also have mucus production. So that helps to trap the microbes, potential pathogens. And then that allows for more interaction between antibodies and also immune cells so that they can end up handling those potential pathogens or microbes. But even if we look at the reproductive system, we're going to have production of mucus and a lower pH. And again, it's all meant to help to prevent microbial growth. So again, one of the big hallmarks of anatomical barriers in preventing infection are acidic pH. There's also enzymes that are present to help break down material and binding proteins. And then we have these antimicrobial peptides. And I'm just including in table 4-2. Again, it's not expecting you to memorize this, but think about maybe you have a favorite protein or maybe a favorite location, and so these different antimicrobial proteins and peptides play an important role in maintaining that homeostasis, making sure that that area is microbial free. And so I'm just going to point out to the second row to the bottom on this table for looking at proteins. We have our surfactant proteins. So in the respiratory tract, again, we're going to have mucus secretion, but we also have these surfactant proteins that are present. And so they help to promote phagocytosis. So for individuals with cystic fibrosis, they don't produce the surfactant proteins, and so this causes them to be more prone to respiratory infection. And so just to link this to like a human disease. And so peptides are just shorter proteins, and so there's examples of peptides. I would say defensins are probably the most popular, widely studied, just because they're kind of redundant in that they're found in multiple locations. So there's defensin A and beta, but they're found in the skin, mucosal, intestines, respiratory, urogenital tract, and so they're found in all these different locations. And just as their name implies, they're a defense mechanism that we have that helps to kill pathogens or disable viruses. So if these barriers, both physical and chemical, end up not working, so if there's some kind of break in it, then that's where the cellular components of the innate response will step in, because they're going to have receptors, and these receptors will recognize these pathogen-associated molecular patterns or microbe-associated molecular patterns, so MAMPs or PAMPs that we talked about earlier. And so they'll recognize those molecular patterns to indicate that it's not self, and then they'll step in and respond. Also at the same time, as I mentioned, if we have aging, dying, or damaged cells, they can produce DAMPs, damage-associated molecular patterns. And again, these are kind of like danger signals. They signal eat me, and so our own cells can communicate to these innate cells. And it's through these pattern recognition receptors, or PRRs, that are on the immune cells that are able to detect our MAMPs and DAMPs in order to then clear out damaged cells or potential pathogens. Okay, so a lot of acronyms there. So I would highly recommend, if you find it useful, keep like an acronym. Who's the players? What are the abbreviations I have to know for this unit, this chapter? And so definitely, again, PAMPs or MAMPs, and then DAMPs, and then PRRs. So you'll see those kind of throughout, and we have more coming. So you'll see those throughout this unit in the semester. And you want to be able to kind of translate them. And I do try to make sure that when I use abbreviations that I do say what they are as much as possible. So when we look at pattern recognition receptors, or PRRs, there's a wide variety of PAMP ligands. So probably most studied is the toll-like receptors, or TLRs. So there's a number of different monomers that will bind together, so they actually dimerize. And so there's lots of different binding patterns that can occur. And so there's multiple TLRs. And so each TLR binds slightly different ligands. So different molecules, different patterns, molecular patterns from pathogens. And we'll look at a little about that in our journal club paper, looks at different toll-like receptors. Okay, we then have our C-type lectin receptor, so CLR. It's not calcium, lime, and rust, but it's going to bind lectin. So some bacteria produce lectin, and so it can use this lectin. So if it senses the lectin, it can use that. And later on, we'll talk about complement. And so one of the complement pathways uses these lectin receptors. So usually, if you've taken a microbiology class, you might mention lectin, but that's it's like a paragraph in the textbook, or a couple sentences. And so we potentially will talk about it a little bit more than a microbiology class, but the focus would be our toll-like receptors. We then have RIG1-like receptors, or RLR. And then we have NOD-like receptors, NLR. Okay, so these are all different. They have different ligands that bind to these receptors, but these receptors would be found on innate immune cells in order for them to recognize and then undergo phagocytosis. So once we have receptor ligand binding, we have these activation of signaling pathways, and this will end up contributing to cytokine production, inflammation response, and again, phagocytosis. So we're going to end up talking a little bit more about these receptors. So one of the kind of hallmarks of these receptors is that they have these leucine-rich repeats. So you can see that on that extracellular domain there on the left, how it's got that hook. And so these are just there's lots of leucines in there. So if we were to look at a gene and see that there's, this pattern where we have leucine-rich areas, that would tell us that maybe this is, a pattern recognition receptor. Okay. They also have an intercellular domain, also in the transmembrane section. And on the right here, what we're looking at is TLR, and as I mentioned, they dimerize, and so when they dimerize together, they can have different pairings. And so on the top, we have TLR1 and TLR2, and then we have this lipoprotein peptide that's binding in that groove that's created. And then if you look below that, we have TLR3 actually creating a homodimer, and it's actually going to be able to bind double-stranded RNA. So depending upon which monomers come together to form the dimer, that indicates what binding sites are available for these MAMPs, and then that would, and they can bind to different areas, so what binding sites would be present. So as I just mentioned, with toll-like receptors, we have to have these dimers that are formed so that these extracellular leucine-rich domains are then going to be able to bind the PAMPs or DAMPs. Definitely when we're talking about our journal club paper, this is a very useful figure, the one on the left here, just showing the different binding dimers that you can get for the TLRs and then what they're binding. Also, what's unique about TLRs is that some of them are found on the plasma membrane, and then other ones are found inside in like endosomes. So it means that if we have internal microbes, so if an immune cell is actually virally infected, they can actually still sense. And so you'll notice that for the ones that are found internally, like TLR4, TLR3, TLR7, TLR8, and TLR9, these are homodimers, but they're going to recognize different portions of viral components. So if we have a virus, viruses are going to be made up of DNA, RNA, proteins. So just to clarify, viruses are either DNA viruses or RNA viruses. But if you are if you're a DNA virus, you're going to have to make RNA. So there would still be RNA, viral RNA around. Okay, again, just wanted to clarify. So as you can see, your TLR4 is going to recognize your viral proteins, where your TLR3, 7, and 8 are going to recognize RNA. And there's different forms of the RNA, double-stranded and single-stranded, depending upon what type of virus it is. And then we also have TLR9 that's going to recognize viral DNA, right? So again, it just gives us this wide repertoire of what could be recognized by our innate cells. And then on the, cell surface, on the plasma membrane, we have the TLRs that can recognize bacteria. So TLR4, if it's on the surface of the cell, can recognize gram-negative bacteria. So you might remember, if you took microbiology, that this is like Salmonella, E. coli. And then we have TLR2 and 6 that are going to be able to recognize gram-positive bacteria. So this would be like strep, staph, bacillus. There's lots of other bacteria. TLR5 is able to recognize bacterial flagella. And then just looking at these. So then we also have ones that are specific more to like toxoplasma. And so TLR11 and TLR12. Okay, and again, just to point out on the plasma membrane, some of these are heterodimers, so we can have TLR2 and 1 coming together, or we can have TLR2 and 6 coming together. So they're not always homodimers. So maybe you don't find that figure beneficial in the last slide. And so there's a table. So there are 13 different TLRs, and again, they can come together as homodimers or heterodimers. And so, again, I'm not expecting you to like memorize which TLR recognizes what, but you should probably have working examples. And again, what's really important is understanding that TLRs can recognize a wide range of different ligands, different molecular patterns that are coming from microbes. So in our journal club paper for this unit, we'll be looking at TLR9, just to, cat's out of the bag, because the paper looks at CpG on methylated dinucleotides, and so it does look at a number of other TLRs. So it looks at different ligands and whether you get activation. So it's actually a really nice paper to link this because it's kind of like applying what you're seeing here. Now, when we look at the signaling pathways that are activated by TLRs, common pathways would be NF-kappa B, so that transcription factor NF-kappa B gets activated. Also interferon regulatory factor pathways, and then MAP kinase pathways, which lead to downstream transcription factors such as AP1. And so some of these may sound familiar, other ones may not. Really, when we look at downstream, we're talking about activation of cytokines, it also, which will allow for kind of the signaling to other immune cells to come to the location. We can also get it so that the cells then will also it also initiates the cells migrating to lymph nodes and other secondary lymphatic organs. So again, just in case you're wondering, what do these pathways look like? These figures from the textbook just show two of the different TLR pathways and the adapter proteins and activation proteins and transcription factors that are involved in the pathway. And as I mentioned, if we look at the downstream effect, we're talking about cytokine production and antimicrobial. So again, I'm mounting that defense. Okay. So the next receptor we'll look at are the C-type lectin receptors. And so again, these are going to be important in activating the innate and inflammation response. So generally these are going to recognize carbohydrates, which are found on fungi, viruses, microbacteria, parasites, and allergens. So we have some carbohydrates that are unique to us, and they have carbohydrates unique to them, and so it's recognizing these patterns that are found on microbes. And then we trigger the pathways, and that leads to transcription so that we get the productions of cytokines, pro-inflammatory cytokines such as IL-1 beta, TNF alpha, and IL-23. And then we have our nucleotide oligomerization domain receptors. This is the NOD receptors, NLR. So these get activated also by the PAMPs and DAMPs. So NOD1 and NOD2 will bind, and they're able to bind peptidoglycan, which is found on bacterial cell wall, and some other components that are found in bacteria. And this induces expression of antimicrobial proteins and peptides. So the NOD-like receptors are able to initiate autophagy. And so what happens here is that it causes the cells to fuse lysosomes to bacteria within them, and so that ends up killing the bacteria. So you end up getting these autophagosomes being produced. So auto within, phagosomes. Phagosomes are vesicles that contain lysosomes and bacteria, pathogen. And again, the lysosomes, I don't actually know if I've mentioned this, but lysosomes have digestive enzymes, and so those digestive enzymes end up killing the bacteria. Okay, so this is like an internal process. We have AIM2-like receptors, or ALR. So these are going to be cytosolic receptors that would be able to bind bacterial and viral double-stranded DNA. So these have to be internal pathogens. So again, this gives us a way. We have TLRs on the surface. We also want to make sure that if we're internally infected, that if our individual cells are internally infected, that they have a way. And so the ALRs are a way to do that. Okay, and again, it's through that binding of those ligands, like the double-stranded DNA that's viral. These activation of this receptor initiates inflammasomes to be generated. Inflammasomes end up generating IL-1 beta and IL-18 production. We have a journal club paper that we'll looking at inflammasomes later on in the semester. So we then have RIG1-like receptors, or RLR receptors. So these will be able to recognize double-stranded RNA that's viral. So our RNAs aren't normally double-stranded, and so when there are double-stranded RNA, it's easy to identify that as not being self. And so the RNA is going to end up binding to this helicase domain of the receptor. This will end up triggering signaling pathways to be activated. So our interferon regulatory factors, IRFs, will end up triggering antiviral interferon response. So interferons have an antiviral activity. And then also NF-kappa B transcription factor will get activated. So when we look at pattern recognition receptors and their signaling pathways, they're going to end up activating a large variety of genes. So we have antimicrobial peptides, type 1 interferons, like as I just mentioned, that potentially have antiviral activity. And then we also have cytokines being produced that lead to inflammation, so such as IL-1, TNF-alpha, and IL-6. Other chemokines can be expressed, allowing for immune cells to traffic to that area. And then we also have different enzymes that are going to be produced, such as INOS and COX-2. And so table 4-4 just looks at some of the different peptides and proteins that are induced by signaling through pattern recognition receptors. Again, whenever I'm showing a table, it's not that I want you to memorize the table, but just maybe be aware of some of the information in it. So such as, I've mentioned this semester, we're going to kind of pick on IL-6. So, what cells are producing the IL-6? These are innate cells. And that would end up producing IL-6. And what's the effect that they have? IL-6 helps to regulate the activity. It actually promotes the production of additional new immune cells. It can also produce fever as a response too. So one of the reasons why fever is a common symptom for a lot of infections is because viruses and bacteria are going to have a normal temperature in which they're going to function in, and the fever actually causes your body it's kind of like changing the thermostat. You're no longer at your normal temperature, the ideal temperature for the bacteria or the virus, and so it's harder for them to replicate, and that gives the immune system a little bit of a bonus, a benefit, because they're not able to replicate or to survive as well, and so the immune system can step in. So we have additional other cytokines and chemokines also. So again, if you have a favorite cytokine and you can find it on the table, please feel free to look at that. So that's going to conclude part 1 of this audio lecture. There will be a second audio lecture looking at some of the cellular responses and looking at the processes like phagocytosis and such.